How does the nanodelivery system target cancer cells specifically? The following 4 illustrations and photographs will explain and demonstrate how a system for delivering drugs, human genes, and other agents at a nanoscale, that is, at an extraordinarily submicroscopic scale, has been able to target and destroy cancer cells while sparing normal cells and tissues. This first illustration shows the components of a nanoparticle designed to target cancer cells. These nanoparticles, which are the key component of the nanodelivery system, have been developed to be self-assembling, biodegradable, lipid-based, and bear targeting molecules that home to receptors on the surface of cancer cells when injected into the bloodstream. A tumor-targeting nanoparticle consists of 3 components. A lipid shell or liposome shell encapsulates the payload and is shown in the illustration as a series of connected white circles forming a ring. The payload, which is in the center of the particle, could be, as an example, an anti-cancer drug or a therapeutic agent composed of nucleic acid, such as a section of DNA, an actual human gene, or alternatively, a piece of RNA. A human gene known as p53, which suppresses tumor growth, has in fact been used in government-approved human studies and has not only demonstrated it is safe and effective, but that it also makes cancers more sensitive to other available therapies. Various special forms of RNA have also been tested and again shown to make cancers more sensitive to additional therapies. The third component is the targeting molecule and is depicted in the illustration as a series of blue diamonds on the nanoparticle's surface. The targeting molecule recognizes a receptor that is known as the transferrin receptor that is elevated in numbers on a cancer cell surface but not on a normal cell surface. The targeting molecule is used to seek out tumor cells, either in the primary tumor or in secondary sites where cancer cells have metastasized to other organs. The insert is an actual electron micrograph of a single nanoparticle with a size of approximately 100 nanometers. This is about the size of a virus, but to give you some sense of how small this is, if you simply covered the surface of a United States penny with these nanoparticles, there would be more than 28 billion of them, enough to give every person on Earth several of them. This illustration demonstrates how targeted nanoparticles distinguish cancer cells from normal cells. This is no simple task, as our own bodies are responsible for both cancer cells and normal cells. Even our own immune systems commonly cannot distinguish the two, and commonly do not participate effectively in the process to rid the cancer. As previously mentioned, compared to normal cells, cancer cells overexpress certain surface molecules, such as TFR, the transferrin receptor. Nanoparticles that target these surface molecules can target cancer cells selectively, but avoid normal cells. The diagrams in the red square on the right illustrate this process. In the upper circle within the square, 2 cancer-targeting nanoparticles have attached to a cancer cell depicted in pink. However, in the lower circle, there is another cancer-targeting nanoparticle, but it has no affinity for the normal cell, which is depicted in blue. Once a cancer-targeting nanoparticle attaches to a cancer cell, its payload is transferred into the cancer cell, and if that payload is a therapeutic agent, it will kill the cancer cell. Thus, this technology will selectively destroy cancer cells, while normal cells remain unharmed. One of the most difficult sites to reach with chemotherapy is the brain, because the so-called blood-brain barrier does not permit adequate penetration of many chemotherapeutic agents. However, these tumor-targeting nanoparticles have the capability of reaching brain tumors because of their ability to pass readily through the blood-brain barrier, and have eliminated both primary tumors and metastases in experimental studies. When the tumor-targeting nanoparticles are administered intravenously, they circulate through the bloodstream using their targeting molecule to find cancer cells, either in the primary tumor or in the other organs to which they have migrated. Thus, unlike current cancer therapies, this delivery system not only has the ability to carry cancer-killing agents all over the body, but is much more precise in its effects, simultaneously expressing increased efficacy and reduced toxicity. The following microscopic picture demonstrates the specificity of this delivery system for cancer tumors and how normal cells and tissues are spared. In this experiment, the payload was not a cancer-killing agent, but rather the LAC-Z gene, a so-called reporter gene which is used for identification purposes. When cells take up this gene, they appear blue when viewed in the microscope. In this case, tumor-targeting nanoparticles containing the LAC-Z gene were delivered systemically into an animal with the tumor. The reporter gene showed up in a metastatic lesion which had invaded the normal muscle. Only the tumor tissue expressed the blue color from the LAC-Z, suggesting that the reporter gene in the nanoparticle was delivered only to the tumor tissue, bypassing the normal muscle cells which are adjacent to the cancer cells. This final photograph shows the actual results of treatment for metastatic melanoma in a mouse model. Nanoparticles containing the therapeutic gene p53, along with the standard chemotherapeutic agent, were used to treat a mouse melanoma with a propensity to metastasize to the lungs. Mice in Group D received the complete treatment. Groups A, B, and C were the control groups, as will be described. The photograph shows typical findings in the lungs for each of the 4 groups, where the presence of melanoma cells results in a black color. The mouse in Group A received no treatment, and the lungs were filled with melanoma cells. The mouse in Group B was treated only with CDDP, a chemotherapeutic agent often used in the past as a first-line treatment for melanoma, and showed significant improvement. However, 1 week later, this mouse's lungs would look very similar to the one in Group A, because the chemotherapy is not that effective in eliminating the melanoma, but only in slowing its growth. The lungs of the mouse in Group C are those of a mouse treated with an untargeted nanoparticle followed by CDDP. The untargeted nanoparticle contains the usual p53 gene, but the targeting molecules on its surface have been intentionally left off for this experiment. At this stage, the benefits of treatment appeared significant, somewhat similar to the mouse in Group B. However, 1 week later, the lungs in C will look very similar to those in A, again suggesting that this combination treatment lacking an active targeting molecule simply slows tumor growth similar to chemotherapy alone. As typified by the lungs of the mouse in Group D, mice treated with tumor-targeting nanoparticles plus CDDP resulted in longer-term elimination of the melanoma nodules in their lungs. In many similar experiments, the combination of tumor-targeting p53 plus a standard chemotherapeutic agent improved significantly survival of mice in this melanoma model. Often, 50 to 60% of these animals survived tumor-free for many months before eventually dying of old age, yet tumor-free. When this system is adapted for human use for many different types of cancer, different payloads, higher dosages, and/or different combinations may be necessary to achieve the highest rates of cure.